Alright, from outside, now we're doing the lecture from inside Quest today. So, we're going to talk about the cell cycle that we talked about in the last lecture, but we're going to talk about its relationship to cancer. So, what factors affect cell division It's going to be our first part of the question. And we'll say, how does this relate to cancer? Oops, cancer. Alright, and this is kind of a nice time to do this lecture because this is Breast Cancer Awareness Month, and though I think if you're going to start with any cancer, we should kind of just say it should be just Cancer Awareness Month. There's so many of them. Alright, so there's a couple of different factors that lead us to cancer. So cells within an organism's body divide and develop at different rates. So you could think about that. You don't really develop a whole lot of new neurons, which are the cells in your brain. Brain cells are real slow to develop. Whereas skin cells, you're losing those things every other day. So it's not really that big of a deal, but there's different rates. So the brain is real slow, and then maybe skin cells are quicker. So cell division is controlled by what's called anchorage dependence. It's that the cells need to be in contact with a solid surface to divide. So they have to have kind of a foundation. They have to have something to sit on if they're going to divide. They don't just do it all willy-nilly out free and floating. They have to have a solid surface to divide. Density-dependent inhibition, it just means that when it's crowded, cells stop dividing. So if a cell is, here's like a cell maybe, if it's touching on all sides, and then it's got something down here that it's attached to, well then it's going to say, I don't need to divide. I'm good to go and I'm sitting here. There's no need to divide. So that is density-dependent inhibition. It is inhibited from dividing if the density is high enough. Some other things that we need are we need some nutrients, and then we need some growth factors. And growth factors are just some proteins that help us stimulate division. Okay? So here's sort of a look at anchorage dependence here. It's just if we have a solid surface here that we can grow on, then those cells will work together and make a kind of foundation, and that's where they'll be at. If we were to remove some of the cells, then that would cover, because at this point we have density, let me get my thoughts straight here, we have density-dependent inhibition. Okay, so we have the density dependent, so it means everybody's touching on all sides, so they don't need to grow anymore. If we did remove some of those cells, it would trigger some of the cells around it to say, oh, we should divide so we can fill this up. However, with cancer cells, instead of just forming that one single layer, they're actually going to form clumping layers and overlap and just kind of make a general thing that looks like this. That's actually how we start to develop tumors. Okay, let me just say this. I just wrote an email about some tutor stuff. This is hilarious. So apparently those two words are too close together that my brain is still on tutors instead of tumors. This would be tumors. My apologies. Okay. So here's sort of another thing is that cultures of cells suspended in liquid, that's kind of where they'll be at. They just won't really divide. This is one of those things that on the quiz we're looking at, well, the HeLa cells and things like that. So most cells don't really divide outside of a body. They don't have any reason to. However, if we do add some growth factors, it will stimulate division. It will cause them to start dividing, but that's the addition of stuff. So we actually have to work harder if we want cells to grow outside of a body. That's kind of the key. Okay, so some other factors here is that the cell cycle control system is a cycling set of molecules in a cell that triggers and coordinates key events in the cell. So checkpoints is the big thing. There are checkpoints in the cell cycle. They can either stop or signal something to proceed. Okay, they're just like little checkpoints. So there are 3 major checkpoints. We have G1, which allows entry into the S phase. Remember, that's our synthesis, our DNA replicating phase, or we're making copies of the genetic material in that phase. It causes the cell to leave that cycle, entering a non-dividing G0 or G0 phase. 2 is that we've got a G2 checkpoint, and then we have an M checkpoint, which would be for mitosis. So research on the control of the cell cycle is one of the hottest areas in biology, and this was a couple years ago, but still this relates so much to cancer that there is, if this interests you at least a little bit, this is a good area for research. Okay, so here's the first thing, is that we can go through the G1 phase, growing cytoplasm, getting a little bit bigger. Well, if we enter this checkpoint and it says that we're not ready to divide, we can enter this other phase called G0, which is sort of a non-dividing kind of, the cell looks normal. That's our normal thinking of a cell. It's got spaghetti noodles in there and it's not doing a whole lot. That's actually probably where most of your brain cells are sitting at, is they're sitting there at the G0 phase, like, I don't need to divide, I have enough, everything's good, okay? But if we get a whole set of growth factors, we get some signals, there's a whole signaling pathway, if we get those together, it'll actually trigger it and say, okay, well, we're ready to go through the G1 checkpoint. So it'll move into the S phase, which will then move into the G2 phase and get a little bit bigger. But then we've got another checkpoint at the M phase at mitosis to say, okay, did we complete the S phase? Did we copy DNA correctly? Yes, we did. Did we grow large enough that we can make this happen? Yes, we did. Okay, so we can proceed. But there are a lot of steps along the way because we don't want this to get messed up. The thing is, if a cell gets messed up, not that big of a deal. We just kind of destroy the cell and get a new one. It happens all the time. It's called apoptosis. All right, so connection here is that cancer currently claims the lives of 20% of the people in the U.S., so it's 1 in 5. Cancer cells will actually escape controls of the cell cycle. All those little things, density dependent, all the checkpoints that we learn, cancer's just all like, no, I'm not doing that. And it doesn't. It doesn't follow the rules. So cancer cells divide excessively and invade other tissues in the body. Let's see what that means. Okay, so a couple of things. The tumor, not a tutor. Thinking about emails. All right, so the tumor is actually a mass of abnormally growing tissues within otherwise normal tissues. So these are tissues that just aren't doing the normal thing. They're growing abnormally. If we have a benign tumor, it actually remains at its original site and may disrupt certain organs if they grow large enough, but it still stays within the same area and location. So it's at that original site. Malignant tumors, however, can spread to other locations. Okay? And that's what we call metastasis, or when a cancer is metastasized. Okay? So an individual with a malignant tumor is said to have cancer. Okay. So here's kind of what we might have, like a primary tumor formation, is that it's staying within its original location and it's just a mass here. So this might still be benign at this point. As long as it stays within the original tissue and it stays so you can actually kind of cut it out fairly easily, it's benign. Okay. Though it can begin to start to grow out and invade other tissues. That's called like a local invasion. And you might have a little bit of metastatic stuff, but it's staying within its same area. However, we have true metastasis when these things start spreading out and using blood vessels. So the whole thing is it starts to use blood vessels in order to move to different spots in the body. So that's true metastasis. So cancers are named according to the organ or tissue in which they originate. Okay, so colon cancer, and we'll go like easier names. Colon cancer would start in the colon. We have lymphomas. They start in kind of the lymph nodes and lymphatic system. You might have, there's some blood cancers. Even the breast cancer is starting in the breast tissue. So those are some of those examples. Some of the ones we talk about, carcinomas, originate in external or internal body coverings because I lost the word on that one, but there are these kind of, God, there's a name for the external and internal coverings. But either way, that's where our carcinomas start. Leukemia originates from immature white blood cells. Leuc, we determined, was white, so it's from white blood cells that within blood or bone marrow. So leukemia is associated with that. So they're named on where they start. Okay. Localized tumors can be removed surgically or treated with concentrated beams of high-energy radiation. Metastatic tumors are often treated with chemotherapy. Okay. It's increasingly possible to personalize cancer treatment, and we do that by sequencing the genome of tumor cells and tailoring treatment based upon the tumor's specific genetic profile. So it just depends on the cancer. And there's lots of things, there's lots of research out there. More than 100,000 Americans will be stricken by cancer of the colon this year. Colon cancer is one of the best understood types of cancer, and it illustrates an important principle about how cancer develops. So we can look at that one. I'm not going to spend so much time on this one. I'm not super worried about looking at like the oncogenes. Onco just means cancer, and then so it's a cancer gene. But we can just kind of look at the picture. Here's the idea if we have colon cancer. So there's a few cells that have increased division. So there's activation of this cancer-causing gene, which causes the cell division to grow unchecked. Okay. We'll have tumor suppressor genes which are activated or inactivated, because normally you have these tumor suppressor genes which actually suppress or inhibit or reduce or say no to tumors. But those get inactivated, and so we'll end up developing a small polyp, which is kind of like a tiny tumor, so a benign tumor. However, if you get a second tumor suppressor gene that's inactivated, then we have growth into a malignant tumor, and you can see this one tumor has turned into something that's leaching into blood vessels and linings, and it's starting to spread out and move. All right, we're almost done. I'm not taking a break necessarily in this one because I think that we can finish it here in the next few minutes. But after heart disease, cancer is the second leading cause of death in most industrialized nations. So cancer can run in families if an individual inherits an oncogene or a mutant allele of a tumor suppressor gene that makes that cancer one step closer. But most cancers cannot be associated with an inherited mutation. So that means most cancers aren't inherited. And cancer is near and dear to my heart, and it's something that I pay attention to, as both sides of my family has had cancer. So I think that'll be the little Easter egg on this one. That'll be part of it. You know what? We'll do two parts of the Easter egg this time. So big thing here is that most cancers are not inherited. So that's part one, is that you got to write that. Next thing that we're going to talk about is carcinogens. Okay, so carcinogens are cancer-causing agents that alter DNA. Most mutagens or substances that promote mutations are carcinogens. Okay, carcinogens, cancer-causing agent. Okay. Two of the most potent carcinogens, mutagens, are X-rays and ultraviolet radiation from sunlight. So spending too much time out in the sun. All right. One substance known to cause more cases of cancer than any other thing is tobacco. So here's the thing. Part one is you've got to write that most cancers are not inherited. The second part is to list me a carcinogen. This one in particular hits me close to home, so this is why I will continue to tell you that smoking is not cool, and don't you dare think about smoking. Okay, if you do, I will hound you for life. Okay, because this one from 40 years of smoking is what took my father. Okay, and that one hits me right close to home. So on behalf of my mom and behalf of my dad, I must tell you guys to not smoke. Avoid it. It's not cool and it's not worth it. So there's my public service announcement for today, is that I have to throw that in there. So more people die of lung cancer than any other form of cancer. My dad in particular had a really just aggressive form. By the time they spotted the cancer to the time that he died was 6 weeks. So the cancer from tobacco can be really aggressive and fast moving. So although most tobacco-related cancers come from smoking, passive inhalation of secondhand smoke is also a risk. So just kind of be careful where you're at. Tobacco use, sometimes in combination with alcohol consumption, causes cancers in addition to lung cancer. So, all of those things that we call kind of sins do have their sort of possibilities of consequences. Okay. So just to give you a list, some risk factors, so prostate cancers, dietary fat, breast cancers, estrogen, lung cancers, tobacco smoke. Though we can go through some of these other ones, but urinary bladder and kidney cancers, both from tobacco, pancreas cancer, tobacco, oral cancer, tobacco. Okay, let's see what else. There's some other ones. Stomach cancers, tobacco. So there's a lot in there for that. But these are some possible types of cancer, kind of where they originate at. And so there's a lot to those. And some of those risk factors, which a lot of times just kind of stay out of the sun and don't smoke stuff. That's pretty much covers it. Okay. So with that, that's the end of this particular lecture for cancer and cell division. So if I am seeing you in class, I hope that we're getting this finished and worked on so we get an idea of how what we're learning relates to something else. We can certainly look at HeLa cells as well, because that plays a huge role. But, if nothing else, get those things down, take a look at it, make sure that you have an idea, and we'll review everything in class before next week with our test. So if you have any questions, shoot me an email.